Hello, welcome everyone to today's SKC Science and Technology Webinar series. I'm Deep Medhi, organizer of this webinar series. It's my pleasure to have Dr. Rubul Mout from Harvard University and Boston Children's Hospital. And he's going to talk about from protein molecules to stem and immune cell. So what does bottom-up engineering means in that context? So obviously it's in the biofield, and how does engineering plays a role there? Dr. Mout is a very accomplished researcher. He grew up in Assam. He's author of 2 books in Assamese. One is actually a memoir, one is a collection of short stories in Assamese, and he has co-authored 35 research papers so far, and inventor of numerous technologies in gene editing and protein design. And he did his PhD from University of Massachusetts at Amherst, which is actually not too far from where he is currently doing his serving at Harvard University in the capacity of a research fellow. So with that brief introduction, I'm going to hand it off to Dr. Mout to share his thoughts, and it is going to be like a fireside chat. What we said, he's going to have some slide, and I'll ask him question to clarify. And even for me, many things he's going to talk about is going to be new. And as an audience and attendees, you can put a question in the chat. Depending on how it is, I'm going to read it. And of course, if you want to unmute and ask it directly, just flag me. I'll be happy to have you talk through the questions you may have. Please go ahead.
Thank you so much for inviting and letting me speak here. So this is when I sort of talk to different audience for various reasons. As you mentioned, I also write. So that's a different language. Writing for people is it's a different spectrum of like how you use language, and then speaking to students, young students again that's a different kind of language you have to use.
Right.
And then and then you speak, give scientific talk and like to specialists. That's a different kind of language you have to use. So I'm still learning. I'm still trying to get used to all different kind of languages that requires. So today this is going to be on the science side, not too much of public speaking, but again I'll try to make it as easy as digestible possible. And then so that's one thing. And then the second thing I wanted to say before we move on is that so last week when I was asked to submit an abstract for this, so I was thinking I'm going to talk about protein molecules, designing protein molecules from scratch, that nature did this for 3 billion years now. And then I will move on to actually use some of those protein molecules to engineer cells, which is in a larger scale, like molecules and then cells, right? And then yesterday night when I was making my slides, I thought, I'm trying to cover too much, and it's just two different worlds. And
right.
And from my experience, I can tell you, like the folks in protein design, probably they would not understand anything in the cell engineering side, and then the vice versa. It's basically trying to bridge two different worlds. I mean, fortunately, I happen to work in the both world. That's good. But then again, for a scientific talk, it's just going to be too much in 1 hour. So so you have to excuse me, and I basically took off that cellular engineering, kept it aside. I thought, okay, sometime later.
Okay, that's fine. We can do another session at another time. Yes. So you want to put it in presentation mode? The slide is not.
Yeah. Okay. Let me put the. That's is it good now?
It's good. Yep, very good.
Yeah. So so I decided I'll talk about only about protein designs, and again this is an incredibly interesting and complicated problem that nature has tackled. As I said, last 3 billion years. So I'm going to talk about this today. Again, only 1 hour, very short time to talk about everything. It's going to be a little bit high level, and so a little bit of work that I did And most of this, this work was done at the Institute for Protein Design when I was at the University of Washington. And so that is that. And as I said, this is going to be a little bit high level. It's not like, I'm not trying to go much into, experimental things and all.
Right, that's fine.
Yeah. So because I'm talking about designing protein molecules, which again, I keep saying that nature built for billions of years. And I thought, we should have some, what protein machines can do. I mean, we all, like the common language for protein is that, okay, we eat protein, right? And then we get energy, right? I'm not trying to talk about the things we eat. I'm trying to talk about these molecular machines that actually do, amazing things in our life. And because of these machines, our life function, right? And so before getting to my the work, I just want to briefly play this video, which is is the video visible now, or is it still my talk?
Video is not showing up yet.
Okay, so I'll do one thing. I'll stop sharing, and then let's actually share the video. And very little also. And this is a widely watched. It's visible now, right?
Yes.
Yeah, this is a widely watched video, and then all the participants, later it's available in YouTube. Later on, you can watch. But just to give a flavor, how complex the protein molecules are, how complex our life processes are. So I thought I would, I would play as a sort of background, whatever. Okay, let's start this. So this is all talking about the inner life of cells. And so you enter into a cell now. So you see all these protein molecules. This is most likely two cells connected to each other. And inside the cell, you have all these different amazing machineries. And humans have about 25,000. And they all do their own jobs in different way. And all these machines come in different shapes. They are like ropes. They try to reach things from one person to another. And again, these are all molecular machines. And some of these polymers inside a cell, these are actin or filaments that work as a road. Molecules can move along this long structure. So this is, I think, most likely tubulin. It's one of the main transport system inside our cell. And these are, so here you see a cargo molecule moving along that road, and which is part of the cells and needed for transporting molecules from one side of the cell to another. For example, things that come from outside to inside or inside outside. And here you're seeing mRNA molecule coming out from nucleus, moving to cytosol. And now another machinery, which is called ribosome, will come and start making new protein machines. So here you would see the ribosome is walking through the RNA. And I think this is a piece of, so that's the ribosome, and it's protein synthesis machine. Yeah. And so then again, you see that the cargo walking through tubulins. So I'll stop here. It's a very quick video. There's a longer version of it. I just wanted to give as I said, I just wanted to give sort of like how complex life is inside the cell, and it's full of machine. And I keep telling, humans have about 25,000 of protein molecules, and they all, all proteins come in different shape and different function, and it's just amazing how all these molecular machines, they're orchestrated together to function life, right? And that was built over 3 billion of years, and in an amazing process. We don't know why it happened, but it happened. And so So this, I mean, most likely now you got the idea that the proteins that I'm going to talk about today, I'm not going to talk about the one that we eat. I mean, we eat the same proteins, but for it in a different way, different purpose. But I'm going to talk about building protein machines that can do new functions into our cells and sort of, like cure disease or do whatever.
So I have one, general question at this point. When you say the word building protein machine, are you talking about creating the new proteins, or you're talking about the whole process of how protein is created?
So it's both actually. We are
Okay.
trying to build new proteins that
Okay.
does not exist in nature yet. So I'll come
Correct.
to that point, like how much is available in nature and
Mm-hmm.
why building new proteins just the way nature built is important. More than now more than ever for curing disease and things like that.
Okay.
So yeah, it's a both
Okay,
understanding
that's
the
fine.
process as well as trying to build it.
Okay, great.
And so this is just recap, one slide presentation of how complex these protein machines can be. Again, as I said, humans alone have in 25,000 proteins, and there are variations and things like that. And more than that, more than 25,000 of proteins. And so here, I just wanted to highlight some of this. In the video, we saw this long, these are tubulin, making roads inside the cell. And then we also saw things walking along these roads. And then we know about, like, we talk so much about the coronavirus. We know about the viral structures here, which is like a soccer ball. And then these are very high level and bigger protein machines. And there are so many tiny, tiny, small protein machines actually, come in different shapes and sizes. And because of their shapes and size, they all do different functions, their own functions. And this picture was taken from a protein database bank. And so where does protein come from in our cell, right? I mean, again, this is most people would already know because we study this in biology. So the proteins come from, so we know that genes are they are the blueprints, right? It is inside the cell. And we know that we inherit the genes from our father, and then cells divide, and every cell have genes, except like cells like RBCs or things like that. And so the genes will make inside a cell. Genes will make something called messenger RNA, and messenger RNA will carry the information, and those messenger RNA would be read by another factory inside a protein called ribosome, and which will basically make this protein. And so that's how the proteins are made inside our cell, and there's no other way you can make proteins. I mean, that's only one way of making proteins. And so we know about DNAs and RNAs, and for proteins, and the way it is made is it's a long stretch of chain. It's a long chain of molecules basically, and they're like they're strung together. Like here you see like different beads, and each beads represent for a molecule called amino acids. And so basically protein is a long chain of amino acids. You see all different colors of beads representing 20 different amino acids. So we have 20 good. I mean, there are like we call canonical, non-canonical. They're not non-canonical amino acids, but these 20 amino acids are conserved among all different forms of life, because life evolved from maybe one like primary whatever, right? And then so they conserve all along. And
a quick question here, that you said there are 20 different kinds of amino acids. Is that for human? I mean, you said it's for human. What about other species? Do they also have the same?
20, these 20 amino acids are conserved among almost all life forms.
Okay,
there are
very
subtle
good.
variants, and so that's why I was mentioning, because life evolved from one primitive whatever, right?
Very interesting.
And so it stays all across species today. And I mean, there are subtle variations, like there are few non-canonical amino acids and all.
Right.
We're not going to talk about that today.
Okay,
but more or less, we have these 20 building blocks, and that build all the protein molecules available in nature today. And you could ask this question: why do you need 20 different kinds, right? I mean, it just the answer is that more you have, more diversity you have, right? If you have to build protein machines with shapes from just one amino acid, you're not going to get different diverse sets of machines, right? But for life to operate, you need a diverse sets of machines, molecular machines. So you also need the building blocks to be different.
Okay.
And so that was the purpose of nature, to have at least 20. And you'll see later how this just 20 amino acid leads to like billions of different kind of protein molecules. And so here again, I'm only showing it's a long string. You could imagine, like, for example, this one, right? It's a long string, right? It's a string of amino acid. You could ask how this long string of amino acid, it folds into shape, and that gives the final function, right? Like, what is the science behind that? Like, what determines this folding of this string of amino acids that will give a shape, and it'll do a unique job, unique function, right?
Right.
And that's kind of one of the biggest mystery of science, and we still keep talking about this. Lots of phrase, once in a while they will say like, oh, we solved a 50 years mystery and things like that. Like recently, like we talked about AlphaFold a lot, but frankly speaking, there has been tremendous advance in science, but also there has been a lot of things that we are still trying to understand. And so one thing that happens is that, so I said, there's all these strings of molecules in a long chain. Usually the regular size would be maybe 100 molecules, like 100 bits in a chain, or 200. So usual size protein would be between 100 and 200 of these amino acids strung together. And so it'll first form what we call secondary structure. It'll fold into a secondary form, and then it'll further fold into a more complicated form, and then it can get even crazier, and that's what finally it gives the machine. So there's this protein folding that happens for this, and it is entirely driven by physics, because it is about, so we are talking about 200 molecules, and in terms of number of atoms, there could be thousands, right? I mean, one amino acid is built from maybe 20 different atoms, right? So we are talking about thousands of molecules we are trying to put together. And so naturally, there's physics behind all these atoms, and they will start interacting with each other. And because of that, it will finally lead to a three-dimensional shape, which will give you the function, right? And the reason why we have...
So that means, in some ways, obviously, because of the physics, that intermolecular attractions and everything, all of those also play in some way at that very, very, minuscule,
absolutely
space.
Absolutely. They just trying to, like, this is why in my abstract I said, nature built this, but we humans are trying to build it. But the reason why humans fail is that there could be at least thousands of different kind of interactions with all these atoms and molecules, right? And being able to simulate that at that scale, and you could imagine what kind of, like, computer power you would need.
Exactly, right. And so yeah, exactly, trying to simulate even a simple thing takes a huge amount of compute power, right?
Yeah, so we'll come to that. We'll touch up. Obviously, we are not going to talk everything. And so one of the things that happens in the folding of a long string of amino acids into a three-dimensional shape protein is what we call the energy landscape. So what I mean by energy landscape is that again, like, this is the example. If you have this long stretch, most I'm holding from both ends. And because it stays as a long string, but if I leave it go, it'll just, like, by itself, it'll fold, right? And why it is happening? It's all different tensions and different. So what it is trying to do is it's trying to go to the minimum energy, right? Because it does not want to stay at the very high energy level, right? It tries to go to the happiest mood, where it would be minimum energy, and it will stay there. So same thing happens for proteins. And when they began in the ribosome, the machinery that reads the RNA and starts making these long stretches of proteins, and they began at a very high energy states. And as soon as they move into the cytosol, and different things happen, and they try to find their happiest state, meaning the lowest energy state. So they will keep going down what we call the energy funnel. They will start going down and down and down to find the lowest energy and the happiest state. And because it is important for every molecule, or everything in this universe, everything wants to go to the lowest energy state, right? Because that's how you stay as a stable whatever species species is a word used for different things. And so that's the protein folding landscape, which is extremely important for both for understanding the protein molecules, how they fold, and also sort of, if you want to build new something, which is you have to walk through this landscape, which makes things way more complicated, because how do you even know all this different landscape, right? How do you even calculate all these different states? Like, I mean, if you take just one like 100 amino acid long stretch, and there could be millions of different energetic steps, right? There could be like even billions, actually, billions of different energetic steps, each molecule, each atom interacting to it. And how do you even like then walk through and find the lowest energy format, right? So that's the kind of problem that it obviously for last, like, ever since we learn about proteins and genes and things like that, biology, last 50 years, I mean, from now it's 70 years, but last that 50 years, scientists scratched their head and tried to understand a lot, but it was making progress, but it was not going much, because it just the lack of competition and lack of like all these resources was so huge, and people are not trying to make even proteins from scratch. And so I just before I move to what we do, I just want to emphasize the fact that so I was telling that there are 20 amino acids, right? And the typical length would be about, let's say, 100 amino acid long, like 20 different kind, and let's say the length of a protein is 100, and that 100 is a very arbitrary number I'm throwing here. It could be 200, and some proteins actually, just like last week I was trying to genetically engineer something which is 5,000, 7,000 actually amino acids. So it is not a, like, 100 is not a usual number, but I'm just taking it as an average number. And even if you have 100, so you could imagine how many different number of proteins you can build from these 20 amino acids, right? It's about 20 power 100. And if you condense that down, it's like 10 power 130. I don't even know what that number is going to be.
Yeah, right.
And so that many different proteins can be made in nature. And because all these species have, shared this 20 amino acids, same alphabet, same language, you could imagine this many can be made. In assuming that there's about 10 million species on Earth now living, and each of them will have about 100,000 genes. Again, this 100,000 is very like arbitrarily picked. It's mostly trying to emulate humans, and with that, you cannot have more than 10 power 12 proteins total, right? And so I was telling that the possibility is 10 power almost 130, but we have only about 10 power 12 in nature. Then what does it mean? So this is a slide I stole from my previous advisor, and so a couple of years ago he wrote this article in Nature that the proteins, all these dots, the white dots represents the archipelago in the ocean, and those are naturally existing proteins most likely, and rest of all this vast ocean is empty basically. And so meaning that this many proteins possible, the whole field, but there's only these dots are made by nature so far. And why is that? And so there are two reasons why is that. So first is that the evolution is very short. It's only 3 billion years, unfortunately. And that's one thing, the nature hasn't got enough time to explore all these possibilities. That's one thing. And then the second thing, as I mentioned before, is that the protein works through that energetic landscape, right? And most likely majority of those high energy states are not going to exist, right? And so what happens is that despite the possibility of all this, the whole ocean filled up with proteins, but we have so far in nature only maybe all this archipelago. And so here's what we we tell people, that we still have the possibility of making rest of it, filling it up. Obviously, it's not going to happen very soon, but at least we can decide to make some proteins that did not exist in nature, and then start using them for functions that maybe we don't even know yet, or even we know, like we want to cure disease or things like. And sometimes tiny proteins actually can do so much, and you cannot imagine. So I'll give one example. In photosynthesis, right? The photosynthesis complex, right? That can capture energy from nature, right? That is only a couple of proteins together. So
I
now you
see.
can imagine, yeah, now you can imagine only a couple of protein, how big of a deal it can
Okay.
do to the nature.
So if you can sort of go to the idea of creating protein design, the one factor, I mean, it may be very difficult to know if it is going to be, lack of a better time, I'm going to call it bad protein versus good protein. So that is also in the design part of it, that's something you have to think about, that what makes something a good protein versus bad protein. I'm not saying there's a clear definition. I'm just saying that that possibility is still there, right? So I mean, so how can you comment on that briefly? So.
Yeah, so in terms of good protein versus bad protein, so there are two things. One is for the existence, right? In the existence of the protein itself, whether it's a good or bad, meaning that whether it has high energy or low energy, right? So that's one thing.
Okay.
And then the other thing is that its function, right? And whether it does something good or something bad.
That's correct, right? Mm-hmm.
And functional part, sometimes it's easy to predict the function because depending. So what we learn from nature, like after seeing, it's like if you see all 7 billion people, and if you look at them, and then if you know their characters, you'll be able to predict the next person comes into the play, right? What he's going to do. It's something like that. So sometimes it's easy to predict function from that angle. But again, sometimes it's the danger comes when proteins are they seem to be stable, but they are not, slightly higher than the lowest. And when they are not very stable, you don't know what they're going to do, because they will interact with other proteins and, start messing up basically things.
That's another very important thing you just said, the interaction with other proteins. It's
That's,
not that it's going to be isolated by itself.
Yeah, exactly. The proteins, again,
that's designed one, it cannot just, no man is an island type of a thing, right?
No, exactly. And yeah, I think I should have made that point, and thanks for bringing that up. I should have made that point very clear, is that all these machines, don't think that they work in isolation. And there's like huge network of regulation. They keep changing conformation, they keep changing their shape and new functions and things like that. We some proteins even have multiple functions. We call them moonlighting function. It's a very it's very romantic term, moonlighting functions. And so that kind of thing, proteins, they work in assemblies. And that's one thing I'm going to talk about today. Just the way we humans work in assemblies, right? Like there are things that we cannot do by ourselves, but if we 100 people get together, we can do things, right? So things like that. So anyway, so that is that. And so again, I already talked about this, like, if you have to build new protein, what can you do? It is just like, we don't know yet what can you do. Like, obviously we can say we can cure cancer, we can do this, we can make vaccines and things like that, but those are very mundane things that we speak from our own experience. But like beyond that, we just don't even know. I mean, think about if there was no photosynthesis and you were asked that like what photosynthesis things can do, how it can change the world. I mean, probably you won't be able to comment on that. So I because I'm going to talk about my research a little bit, what I did. So I just wanted to simplify the whole process. I mean, obviously it's not simple, but you can imagine that building protein is a problem of building house, right? In the way when you build a house, you have different structural elements, right? And you have the foundation, you have the walls, windows, you have the pillars, you have the roofs. And the fact that at the end of the day, a house looked very unique, but there are elements like all these walls and foundations, they're sort of common. They're they have the common structures, in the fact that you can easily turn one house to another just by changing some of the shape or things like that, right? So what I'm trying to say is that all the foundations, all the building blocks of proteins are same, but it's just how you stitch them together finally. And so that's what makes the protein different. And so from that angle, proteins also have very common structures. So here, the first one, it's a spiral shape, and we call alpha helix. And this is one of the dominant elements of proteins, a shape of protein, and they're always from spiral. I'm not going to go into why they form spiral, but there is physics behind that, why the proteins tend to form spirals. And then there's another called beta sheets, and this basically, you see like, like a, a sheet of like different strands of beta strand, people also call beta strands. And so that's a different this another kind of the structure that protein always uses to build their bigger protein. And then the third kind is we call loops, and these loops do not have a very rigid structure, but they sort of connect all these beta strands or alpha helices. And these are three very fundamental the structural element that protein have, and they all, most of the proteins are built from these three elements. Just the way, as I said, the house is always built from roofs and foundations and walls and windows, and so that's the analogy I wanted to use. And obviously, when they start using these elements and things go even more complicated. For example, here this is a globular protein where you have both this spiral alpha helices, you have these green loops, and then you also have beta strands. And so that's a that's a protein that it does its unique function, and then they can also get assembled together. And we just spoke about how protein interacts to each other to do bigger things, and this is a perfect example how 5 of the same protein get assembled. We call self-assembly of these proteins to make it larger things. And then this is an example of virus, how virus, I mean, obviously we all know how how deadly, how difficult life has been, but from artistic side, the virus is built from maybe 120 copies of the same protein that self-assemble to build this large outside shell of it, which is so important for both vaccine and for
this is very interesting. I mean, I like this in the sense that you kind of go from all the way from protein to the virus, what actually goes into that form of things, right? So is it fair to ask a question that's related to saying that you create we talked about good protein versus bad protein. So does it mean that certain proteins that you can create potentially?
No, that's I mean, this possibility is always there, right?
Always
any
there,
kind of
right.
engineering, like any kind of
exactly.
Yep. in the internet world, we have seen all this cybersecurity and all
exactly
this.
So but my point was not to kind of get people scared, but point is that you always
exactly.
you always have to take a chance. You have to take a bold step to new problems. You cannot be scared of what's going to happen in the future. Otherwise, we would not even have the internet to start with.
No, exactly. So, same thing applies for all these technologies.
Exactly,
I mean, like, what can you create? You can create bad and good, right?
Exactly.
And just that, for the good thing is that humans, we live as a society, and we have all this ethical regulation and things like that. Those are in place. So that usually takes care of, if somebody wants to go crazy or things like that.
And those are also very important. Once you create
it's so important. It is
very,
so
very
important. Otherwise, like, one single, right now, what is happening to the world, right?
Right, exactly.
One single person decides something, and it basically destroys, like, it's so many lives, right?
Exactly.
So that has been place.
Okay.
So this, again, I just wanted to simplify how, obviously it's very complex, but also how simple it is, right? They're at the core, there are 3 different kind of structures that all proteins use to build their own machine. And so I just wanted to so this problem was there for a long time, but nobody could actually touch on this problem because it was just so hard. And it was hard because you need understanding of proteins at very molecular level, all these different interactions, and then you need computer skills, right? Good, like really good computer skills, with a lot of resources. And so this problem was first took up by David Baker in 1998 when he started his lab at University of Washington. Basically, he graduated from a lab that went on to get Nobel Prize later. But David Baker, he was a cell biologist, but he wanted to take this problem from computer science angle. And I don't think he had any education in computer science before, but he just like he joined as a faculty to do computer science. I'm telling this story because, like, as you mentioned, you have to take bold step in life, and unless you take a big problem, like recently David, he got Breakthrough Award last year. Breakthrough is like such a big award, right? And David was saying that the danger in science is not tackling small problems, not tackling big problems. It's tackling small problems, and that's pretty much true. In science, you have to take a big problem, otherwise you basically ruin your own career. And so David took that. That was almost 25 years ago, and he had been extremely successful in even he tried and things work now. And so he built up. So first thing he did was he wrote a software that called Rosata.
That means he learned programming. He did not have any programming background probably.
Exactly.
Yes,
and he was an assistant professor. How dangerous to
how
begin
dangerous
something new.
to do that in U.S. institutions. I know exactly what you mean. Okay.
And so he wrote this software called Rosata. Now obviously Rosata is like universally accepted by almost every university, maybe every biochemistry class also in the United States teach about Rosata. And so he began 25 years ago. He put all these all these interactions, whatever understanding is available, whatever physics is available, put into this package of software so that he can start doing calculations. At least he can predict what is high energy and what is low energy.
Low energy, correct.
And
that's very important.
If he can predict that, if he can calculate that, it will at least give him a idea like what can exist and what cannot exist, right?
Right.
And if you get better, over time, you would be able to make new proteins, because then if you can predict like what is stable, what goes to the lowest energy, then you should be able to build new proteins. That was the whole idea. I mean, obviously, again, it's not that as simple, but that was the idea. And so when David started Rosata, again, we talked about if you have to build new proteins, you have to build these 3 different foundational structures: the spirals, the strands, and then the loops, right? And again, building all of it itself has its own problem. How you basically put different amino acids to build all these things, and how you calculate and things like that. But the way he approached this problem is that he would first create what we call backbone. Backbone in a shape of spiral, basically, right? And then he'll assign all these 20 different amino acids so that it makes this backbone stable. It exists there. So that was his approach. And the way he would do this is by rotating he'll let all these amino acids freely move around and interact with each other. And for each move, there would be a calculation. And based on that calculation, you get an energy basically funnel, right? And so because there's so many atoms and so many amino acids in a structure, you will have at least millions of these possibilities. And you calculate all of those, and then you go down to the lowest energy that will most likely exist if you have to make that protein. So that was the approach. And so he built that software. And this software Rosata is about 3 million lines of code now. And Rosata has a huge community, like some 500 people across the globe work on Rosata now for the development.
It's like open source code, basically, right?
There's a huge Rosata common community. There are at least 60 to 7 different labs around the world who actively contribute to the development Rosata now. And that also lead to new tools, something like Rosata Pro and things like that. Yeah. So what did David Baker and all of his people did over the years? And obviously he started from very beginning. He started building all this foundational, the spirals and things like that. And then he moved on, and he went by 2016 he could actually build things like the things that look like virus, and the question that we faced earlier. And he could build large protein machines that is made from 120 different 120 different protein molecules, and it'll self-assemble to form this large virus-like structure. And 2 years later, he was also able to basically have its own genome encoded into these things. So again, people would get scared by these things when you see you can make artificial virus from scratch and things like that. But again, as I said, there's regulations, and there's a good reason to build all this, because one of the things that I'll show later is that developing vaccines, right? How can you build vaccines rather than making a deadly virus, right? Things like that. And so in 2017 I went to work with David. I was very fortunate to work with him at the Institute for Protein Design,
Mm-hmm.
and especially he set up full empire of things, and with groundbreaking technologies that he himself developed. So the problem I was trying to tackle there is that so a lot of these large protein assemblies, virus-like assemblies, have been already made. But one problem was that so how to basically have all these spirals which
Mm-hmm.
do not have branches to hold on to, assemble, as if you have only the trunk of the tree, but you do not have the branches. And so if you do not have the branches, it's very difficult to sort of assemble them in a three-dimensional structure. And my problem was, how do you take all this very rigid, without any trunk, and then actually try to make them into very large structures that can be used for vaccines and things like that. So that kind of problem I started tackling. And so one of the things I started doing is that again, I don't want to go into very technical details. I just trying to give a very high level explanation of. So one of the things I did there was that we, along with my colleagues, we developed different programming tools which can actually allow us to positions all these different helices, so we call alpha helix, different helices together, so that you can stitch them together to first take this very rigid thing and then extend with arms, and then with the help of this arm, you can further assemble them to make in things that looks like virus, basically. So the problem I was saying is that if you do not have these arms here, you won't be able to make this large protein assemblies from these rigid things, because they do not have arms. So you can imagine that, right?
Mm-hmm.
So that's the problem, and then obviously it requires lots of geometric calculations and things like that.
Right.
So that's the problem we face. So we did multiple rounds of designing new proteins using these traces. Here, at the core we have a tetramer, we call, because it has four different proteins assembled together, and then we put these arms, and this is a design in computer, and this is a real experimental. We build this protein, and you can see this with electron microscopy, and you can also see these beautiful rods coming out, and these
let me stop you just for a thing. It is very important. I want to kind of reiterate something you said, which is that although I mean, computing or rather software technology has allowed you to do this protein design and stuff, but you also need some sort of a ground truth, in the sense that you can, the wet lab, you can see that, okay, that's what it is going to be under electron microscope. So that's very, very important. I want folks to, the other thing I want to quickly point out is that the reason you want to do that through computer software to develop it, you want to rule out a lot of things that you want, that's one of the biggest part of it, that you save the, like what I call is the solution space, so that you can then on the wet lab only can go do the ones you want to look at it. Otherwise, you're looking in the ocean, you're not going to find anything, and it's going to take you forever to even do anything. So
so I talked about the different possibilities, right? I talked about a single protein can have millions of different stages, right? And unless you use a computer to calculate that out and try to find what is lowest, you'd be spending 1 million years if you have that much of lifetime.
Obviously. Exactly. So it's saving time, huge amount of time. Just being able
absolutely.
Another quick question: the computing power point of view. Trying to do one of these things, what sort of computing power you need, actually?
So it comes with different level. There's a hierarchy of experiments that you can do, and first building the core, and then you go on, and each level requires different kind of computation depending on how complicated it is. How much is you are calculating, and because it varies, and as I said, there is a hierarchy of computational power uses. It would be from from terabytes to like supercomputer cluster or something like that. My computer, I had a 2 terabytes memory, like MacBook Pro. I could do very simple calculations in my computer.
Exactly.
But again, I have the access to all this cloud space and things.
Correct,
like that.
right. So the supercomputing is very important.
It was very important.
I knew that's exactly what you're going to say, but I wanted to have that be on note for the audience to realize.
Yeah.
That
So 10 years ago, for example, 10 years ago, it was difficult. And
absolutely
things are getting
impossible.
better.
It's unbelievable what is happening right now, because this is one of the things I support at NSF, for example, about the computing power, because knowing that I feel like where the biggest contribution is going to come is in the intersections. I call it at the intersection because of different fields. Because if we can make the computing, people just think, okay, putting just computing power, that's not it. But the software, you have to write it, to be able to run on that. And because you have bigger computing power, you put more complexity to your software code so that you can understand even more minute level of detail about interactions that you could not even imagine calculating 10 years back. So, all of this kind of plays together to be able to understand, inner life, as you kind of said it at the beginning, right? So that's sort of,
No, absolutely, yeah. So this is these are some of the proteins that we build. Obviously, this also look nice, aesthetically, like this, especially this one I like so much. It looks like a flower vase and things like that. And this looks like some kind of wheel, which actually in nature they're exactly like this. There's a protein. This is from scratch, built from does not exist in nature, but nature has exactly like this kind of protein. It's called inflammasome. When different different microbes or different germs invade us, our cells actually respond to that in order to fight that. And so there's a protein complex called inflammasome, which looks exactly like that. And when I built that, I was like, oh my god, nature took billions of years. I built in 2 years.
So you're smarter than nature. That's what we can say.
Well, with the help of all these resources.
Of course, resources are important. Of course, important. Yes.
And so we went on and on, build so many different protein complexes. And this one, obviously it looks very simple here. These are both the computer model as well as what data we get electron microscopy to see how they look like. It basically fits very well. It just looks like what we built. And just in terms of complexity, although it looks very simple here, but this is actually built from 8 different proteins, and they assemble in a very geometric precision that it makes only in that way, which is again with the help of computers and things like that. And then we would build even bigger things like this we call crowns. Again, I'm not going to go into more details, but as you can imagine now, from, as I said, 20 years ago, you cannot even build a single amino acid, put into something, but now you can build this, and this is amazing. 20 years of advancement in science is just so much. And then this is another we call tetrahedral geometry. It's just a tetrahedron, and they just assemble, and these are some experimental data. This is the icosahedron again. This is like the virus, but it is not, and also it has lots of pores. And when we mapped with electron microscopy, it looks like this really beautiful things there. So we made lots of this. Again, you can ask why are you making all these things, right? I mean, again, the point is that making any protein machinery that you want to build, the first step is to build the parts. If you want to build a car, you cannot, I mean, obviously these days we do 3D printing, but you cannot build a car right away. You have to build all the parts, right? You have to build the glasses and the things. And then, so once you have all those, you can assemble to make a car. And why only car? You can make different things when you have all the parts, right? And so this is again similar strategy that we have to build all these parts step by step in a hierarchical manner, so that at the end you can make really big machineries that can help again solve problems. So again, obviously there's no end in research. You can keep going and making things. But I just want to give some flavor about so far whatever we have built, what kind of problems we can solve, right? What kind of real world problems.
Okay.
Right? Because in science that is important. So So this is a protein that exists in nature. This is not a protein that we made in the lab. This is called antibodies, and I'm pretty sure by now almost everybody familiar with antibodies, because antibodies are the proteins that our body used to fight against germs, right? Like in COVID, when you get vaccinated, we make antibodies. Our bodies make vaccine antibodies which can fight against COVID, right? And this protein is made by cell population of B cells. And these proteins are extremely important for our survival, for fighting off disease and things like that. And so one of my when we are developing these tools what happened when we are developing these tools to sort of both computer as well as the way how we are going to test things. So one of my colleague, he was a grad student at the Institute for Protein Design. He took our tools and he took this antibody and he wanted to make something that does not exist in nature. So I'll come to it why he wanted to make that. And so, as I said, in nature, in our body, we have this, the antibodies look like this Y-shaped, molecules, and it's very good at fighting. And so he wanted to take the same approach that we use and use some of the design protein to scaffold so that he can make larger assemblies together. And so he ended up making assemblies look like the virus out of these antibodies. And then you can ask this question, like, why would you like to make virus like assemblies from antibodies, right? So one of the things is that I keep telling people that nature does things. Nature, in evolution, 3 billion years of evolution, nature have done a lot of things. But also, true that nature has not done a lot of things, because again, because of the amount of time nature got. And so throughout evolution, nature made these molecules to fight disease. But what nature did not do yet is that, you can imagine, like, when you go to a war, if you fight alone, you can win, but you can have like 100 soldiers and there's a better chance of winning, right? And so what nature did not do is that nature did not take these antibodies and group together to form this kind of molecule so that they can fight together. And so when Robbie made this using both naturally existing antibodies and then laboratory made proteins and build all these virus looking structures, and he was I'm not presenting this data, but he was able to fight even COVID and even in cancer, he was able to give a very strong protection against a lot of these diseases. And he basically used the tools that we developed.
It's why it is interesting is that in a sense the whole idea is very simple. It came to his mind, and then he said, what if I change this a little bit this way? You think about just taking a small, almost seems like an epsilon step on one side, and but it creates a whole new array of things that you could not even imagine. But it's a simple, right? So
absolutely. And this kind of technology, like a lot of this, the powerful technologies come by slight change and looking at problems from different angle, right? And nature has been doing that, by the way.
Nature has been doing it over 3 billion years, as you said.
I keep telling people that each of us, we are one single experiment.
Right. right.
So Robbie, he published his work, and it actually came in the cover page of Science. And
wow. Okay. Right? That's very nice. Yeah.
The new way of seeing things.
Yeah.
And then the other thing, again, I'm not going to talk too details about this, and I have been keep telling that you can also make vaccines and things like that. And IPD, Institute for Protein Design, actually have been actively working on designing vaccines. And there's a lead vaccine for this COVID. I mean, obviously, we already have COVID vaccines, but the IPD is trying to develop one that is a universal vaccine. Universal vaccine is the one that actually it fights against wide range of germs, right? The whole coronavirus family, right? Not just COVID-19.
Exactly.
So those kind of
SARS-2 viruses can.
Exactly. So those kind of vaccines, it's extremely difficult to build. But with all these new technologies, how you can build protein scaffolds, how you can take naturally existing proteins like the spike protein of the virus and put into all this different forms of spike protein into all these different man-made proteins. it's amazing to see. Again, 20 years, you could not have imagined this, but now it is possible. And IPD is like very actively pursuing, and they're also using, obviously I was there and using some of the technologies that I developed to, further sort of it. And this, the Science magazine wrote an article they call this vaccine,
the
dream vaccines, because the reason why they call it dream vaccine, because it's so modular, and you can like put all different antigens on the surface, and there is now all these protein engineering tools available, and you can just do so nicely, and like build a vaccine against basically to the whole pan coronavirus or things like that. Again, I'm not going to go into too details. There are many papers from IPD published already on this. If somebody is interested, can look into that. So now I'm not sure how much time I have, but I just want to move from, so far I talked about making protein molecules that have rigid shapes, and that gives to function, right? So what we call ordered proteins that has function, right? And so one of the things that scientists did not pay much attention until 15 years ago was that until 15 years ago, everybody was thinking in order for a protein to function, you need to have a shape and some kind of very rigid shape and size, which is fairly true for majority of proteins. You need to have some kind of shape so that it can function something, because it's a machine. But about 15 years ago, 20 years ago, people started, scientists started realizing that there are a lot of functions that do not have shapes, and they still do very important functions.
Exactly.
And so you could ask this question, like, why is that? Why do you need no shape for some functions? And I'll come to few of those, but it's actually last 10 years has been like very intense research going on in this area, how not having a shape is beneficial for certain functions of protein machines.
So before you go to that, I want to kind of point out something important you say, just for the audience, which is that we sometimes get stuck in the way we get trained, and you think, like you said, oh, proteins must have a certain shape. Then you realize actually that's not true, because you are so ingrained in a way to think about it. So it's very important to keep in mind to think out of the box. Basically say that, that's what we saw. Is that always going to be the thing? I think that's where science happens. That's where technology happens all the time, because that's where somebody saw a little bit like Robbie's example you gave earlier, and now you're talking about the shape aspect. You suddenly, but so don't be so ingrained because that's how you got trained in your high school or science book and stuff like that. You suddenly realize that your biology book in the high school is actually not correct anymore.
Yeah, no, absolutely, and especially when we got educated, obviously that is a totally different time point of the civilization we live. And I'm saying it's a very time, it's obviously only like 15, 20 years apart, but things are happening in such a, like, the speed of things happening now is just unimaginable. And, we saw this.
And this is also helped by having the computing technology.
Yeah, computing, and then all this,
everything.
All the convergence of all these different technologies and understanding and knowledge is so much that time thing, it's changing in maybe in exponential scale or logarithmic or whatever you call it. And so so 20 years ago, whatever beliefs and whatever truths existed, it's changing now.
Okay.
So we have to keep updating on those things.
Sorry, you're going to say something about the which are not shaped at all. So sorry.
Yeah. So so going back to proteins that do not require shapes for doing crazy stuff, do maintaining our cells or having the proper functions of our cells. And so when I was working on all this problem, and that it was a very intriguing problem, and I really wanted to sort of give it a try to build proteins that do not have shapes, or at least the overall. I mean, at the micro level they might have shape, but like in the overall thing, it is like no shape, and still it gives some kind of functions. And at the time, so I'll show some slides that what I ended up discovering later. But at the time when I started this work, I had no idea that I would I would go and find something very exciting that never happened in nature. So I'm mentioning this because in science, many times when you start something, you have no idea what parts you are going to take. You start a project, you have some idea, and you get sort of, get very obsessed about it. You want to try it, but absolutely, like, 3 years down the road, you have no idea what you're going to see.
Exactly.
And I think, if there are some young participants, it's very important to realize that, you don't have to have a perfect everything at the beginning.
Right.
You just have some idea, and then you start there, and you will see like new world, basically, which I'll come later, obviously.
Okay, one quick question here from the audience is that, do you also do simulation to see how the protein structures behave over time?
Because they go to lowest energetic state, most of the proteins that we design, and I don't think the time can be incorporated there. And then protein folding is a very rapid phenomenon. It happens in fraction of seconds, basically, or even in nanoseconds. It is not a very slow process unless there's a very, like, almost at the lowest, but slightly upper than lowest energy scale. Those can take things, those things can take longer for equilibrium, to get a equilibration positions. But most protein fold in like very rapid, time point. So time is not very, helpful thing here. And so I was talking about this disordered protein, right? Proteins that do not have any good structure, three-dimensional structure, yet they function. And so those proteins are now known as intrinsically disordered protein because they have intrinsically disorderedness. And so it turns out that intrinsically disordered proteins have huge functions in our cells, and in different ways. Again, like you can read reviews on how many different ways, but it's just so many. And so what happens is that all these proteins, there is a little bit of the spirals there, but other than that, these are all disordered. And if you take this was I took it from Protein Database Bank, and they did a structural analysis of this protein over time, and they saw it is totally floppy and fluctuating. It's just like these things I keep showing. If you didn't want, maybe it will keep changing, and same way it keep changing. And one of the things why it happens in terms of the protein energetic landscape is that for most structured protein, it goes to the lowest energy. For most this disordered protein, they have at the bottom, they have multiple states and multiple
I see.
So because of that, they basically it's very easy just by changing tiny fluctuation in the environment. Again, that is, we are not controlling it. It is nature is controls by itself, like like temperature, pressure, and or I don't want to say gravity, but who knows?
Yeah, we don't know yet. Yeah, right.
Yeah. And so those kind of things. So, and all these things are important for this. And so the kind of, again, I'm just giving few examples of how these intrinsically disordered proteins actually play huge role in biology. And now there's an emerging field to try to engineer this kind of phenomenon that happens in nature to to prevent disease or even cure diseases. And so one of the things that happens in cells is that when cells get shocked, and so they basically try to protect its genetic material, right? It's just like like you might have seen in chicken, like a hen, when some like some birds, some try to take the chicks, and what does the mother do? It try to protect, right? That's an immediate instinct. And so it turns out all the cell machineries have the same kind of same kind of mechanism to protect its own material. So for example, when there's a shock to a cell, and the cell wants to protect its material, right? And throughout evolution, it learned to protect, it learned to have mechanism so that it can protect its genetic material. So one of the things cells do is that they form something called P granules, where the RNA, which is made from DNA, so DNA to RNA, RNA to protein, so they want to protect the RNA right away. And so what it does is it forms this like stress, that people also call stress granule, so that it clusters everything together, just the way all the chicks come together and hide under the mother chicken, right? And so that kind of stuff. So it's amazing. It's very cool how these things happen in nature, and that is often triggered by all this intrinsically disordered protein, because they need to have this random like wrapping around all these RNA molecules so that they can protect it. And then the other things happen, like during cell division, for example. Again, I'm not going to go into details, but there are processes that are so dynamic. You need to keep changing over time. And if those events needs to be changed over time, you can imagine if you have a very rigid protein structure, it's not going to be able to do, because that protein will do only one kind of thing. But if a protein needs to do 50 different things, it has to have the very flexible, right? It needs to have kind of like all these floppy and non-structured regions, so that it can do multiple things. And then there are bad things also happen because of all this intrinsically disordered protein. One of the things happens that in Parkinson's is that some of these proteins, they actually aggregate. They form this, because they don't have good structure, they form aggregates, and then they kill the neurons. And obviously Parkinson is one of the disease that is caused by this kind of intrinsically disordered protein. And there are many other neurodegenerative diseases that are caused by all these proteins. So anyway, so I just wanted to allude to.
No, that's very good. So we're at the top of the hour, so let's see what else you wanted to quickly cover.
Yeah, I have
I have a couple of questions once you finish.
Sure. Yeah, I think I have 3 more slides almost done.
Great, excellent, very good.
Yeah, obviously I'm talking. So so when I was at IPD, and I was very interested by this problem, how to build proteins that do not have structure, yet it can do something. And we don't even know what we can do, but at least I wanted to try it. Let's build something, and maybe we'll find a function, right? And that's how nature did. First you evolved, and then it got the function. And then the idea was to build first proteins that have structures, but then turn them into unstructured, and through different protein interactions. And basically, we use a lot of those floppy linkers that do not have structure, so that it gives that the flexibility and sort of build networks of proteins, just the way it builds network of protein here. Obviously, here we are seeing a cell, here we are seeing in molecules. So build all this network of protein, that what I call system-wide network of protein networks. And so that you have this kind of phenomenon. And I was extremely fortunate to actually get some of those protein work what I wanted to make. And so again, I'm not going into details. So if you have some of the rigid protein structure, it forms very rigid gel kind of, if you take all those protein, they form very rigid gel. And if you distort those cells, they just stay wherever they are. What it means is that the protein do not have this ability to move back and change their shape to get into a new form. But there's some of the proteins that I built. It's not gel. It is actually fluid, which we call non-Newtonian fluid. It has very interesting properties. And if you take this fluid, they are perfectly liquid kind. But if you give a sudden pressure, they become solid. And they become like gels, and you can break into pieces. And this is a liquid, but you can break into tiny fragments of glass-like pieces. But if you remove that the sudden pressure, it goes back to the liquid format. And I was totally mesmerized by this phenomenon, because in nature we have this kind of all these molecules and things like that happens. But with proteins, the things that runs our life, to get something like that, I was really very excited. And so again, I'm not going to explain this because it will take too much time. But again, we did a lot of mechanical studies to show that these proteins, that the laboratory made proteins, actually behave as like as liquid or solid or even glassy solid at different stages of their like. And then so here's a video where I showed that. Turn that up. And this again, usually like other things break, but this one, it does not break. It fluctuates between this solid and liquid format, depending on how much things you apply. And so this was obviously this protein was made in computer, then first built in computer, then made in in bacteria, purified, and did all this experiment. And then I really wanted to see what actually happens in real inside a cell, because as far as I know, there is no such things that in our life form. There's no such protein that can actually change their properties from liquid to solid depending on the external stimuli. And there are different things, like I talked about RNA granules, right? How when you have a sudden shock, it wants to protect the material and it forms those granules, right? I talked about that, but not like something, the non-Newtonian properties of fluid. And so what I did was that I took those proteins and then so this is a mammalian cells, it is a human cells, and when you do not have these proteins and you see only this green, so this is a giant cell basically. But when you take the proteins that I built, and they again form those tiny droplets. And what was most interesting is that these droplets actually behave exactly the way it behaves outside. It behaves as a, like, if you give a shock, it'll turn into like kind of solids. And again, as I said, in nature, to my knowledge, I don't know if there's something like this exists. But now I'm trying to, begin to envision where, maybe you can have new functions with all these new proteins, right? That nature did not have. You can have cells like that respond to, let's say, you give a sudden stress or something like that, heart pressure, it wants to make things like very stiff so that it doesn't move or things like that. In nature, keep doing a lot of these kind of things all the time. Like one example is all the trees in the.
Is that why you call it non-Newtonian? Because it is not expected in the nature? Is that?
It's called non-Newtonian because the properties of the fluid, because
I see. Okay.
it is not a like linear like water, right? Like this water. So water is water. It doesn't matter what you do, it's going to be water.
Right.
But this one, it behaves as a liquid and resting step. But if you give like sudden something, it turns its color. It turns it, not physical color, it turns its properties. So that's why it's called non-Newtonian. Okay. And so this work is actually, I'm at the final stage. I hope I can like make
great. Sounds very exciting. Yes, thank you for sharing it.
Yeah, so it's just I'm trying to tell to people that as I said that when I began this work, I had no idea that I would go on to invent or discover something like this.
Right. You cannot imagine what it is. People sometimes ask me, do you know what you're going to do in 5 years? I have no idea.
And the fact that now we know that this can exist inside a cell, which is great. But even now, if somebody asks me, what are you going to do with all this? I mean, even I don't have the answer, right? This can be like, some other people will come from different field who would need some different requirements.
Exactly. Yeah. So sometimes the discovery in itself is important because somebody may find a use for it, so that's
absolutely.
Yeah.
Absolutely.
So they will figure out once you do that, so I mean, we cannot do all of it together, ourselves. Other people say, hey, I can use this way. So very good. Excellent.
So so that's pretty much. And as I said, I obviously now I work in a totally different spectrum of the whole thing. And but I'm bridging, I use a lot of these de novo design proteins, and then now I'm trying to build cells and engineer cells. Which today I'm not. This is my final slide. I'm not going to talk anymore. It's just because this is a totally different story. It requires different language. But like now you can imagine, like you can take all these proteins that made in the laboratory. I mean, obviously you can do so many things. You can build vaccines, cancer, blah blah blah. But you can also like do a lot of things that like, for example, I am interested in engineering stem cells and hematopoietic stem cells, which are derived from bone marrow, and they lead to the formation of all these different defense cells, all these different basically cells that defend our body. And it is so exciting sometimes. I feel like it's just like there's so much of excitement what you can do with all these things, and I think I'll end with that.
Oh, this is fantastic. Very, very. I really appreciate you sharing the all the exciting stuff that's coming up. So I have a couple of general questions now. One is that I'll kind of step back to our educational training for a second. I mean, you are obviously originally trained as a biologist, and then.
So can I stop sharing? Or?
Yeah, please do. Yeah. You trained as a biology, but you realize that's down the road that you needed to learn some software programming and stuff like that. So do you have any thoughts for the audience that how without overwhelming anybody, saying that why it is important to learn different set of skills? Do you have any thoughts?
Yeah, so one thing I mean, obviously there's no like one single answer to that question, right. And it all depends on circumstances and your need and your enthusiasm to do things, and they're different players. So one thing I always think that my education was chemistry. I did my undergrad in chemistry. My master's was in inorganic chemistry, about like atoms and things like that. And then today I only talked very small fraction of my research. Basically, I only talked 2 papers out of 35 papers, right? So it's kind of my rest of my work. I have done work in gene editing, genetic engineering, genome engineering, things like that. Now stem cell engineering and all this require different skill sets, as well as different sort of knowledge and language that I have been talking about, right? Like when you go to hematopoietic stem cell, it's like totally different vocabulary, it's totally different world. And so one thing I found is that, and these are like the thing that you do, and then all the tools that you said, like computer tools, different resources, right? Knowing what to use and what kind of technologies, like cryo-electron microscopy and things like that. Right? So all of these things, I think it comes along with your, based on your need and things like. And as long as you have resources available where you can explore, I think it's fine. The one problem with students at very early career, because if for them, I think it's more about not having the resources available for them to learn it.
Correct, right.
So I think it's difficult to compare. But one thing I always tell people, that you don't need to know everything.
Correct. Exactly.
Times,
right.
Like for example, I talked about non-Newtonian fluid things, right? I mean, like I knew nothing about all this.
Right.
I just accidentally,
So the point is that it's better to know some things well, to fundamental stuff, in your like your chemistry you talked about. Get it well, and you pick it up as you go along. Be a kind of a lifelong learner, sort of.
So I think what is most important is to build your platform so that you can have resources.
Exactly.
And I think I keep telling students, right, work hard so that you can get access, because there's so much competition, especially in India, right? I mean, you can claim like I'm super smart and blah blah blah, but like if you cannot get access to the resources, there's no use of that. And you have to get access. You have to get access to all the resources, unless you won't be able to do anything. So.
Right. Very good. All right. So anybody else have any questions from the audience? You can unmute yourself and give you maybe a couple of minutes. It's a little bit long already, but still we want to give some anybody who want to ask a question an opportunity, obviously, before we close today's session.
How do you know that which proteins are useful for us or which are not?
So that's a good question. And usually a lot of proteins that we make, we have a purpose to make it, because also, like, the reason why many times we need to have purpose, at least some partial purpose, if not everything, because we have to justify to all these grant agencies, tell them that why we are making, otherwise they won't give us money to even work. So we always have some purpose, and our motivation is driven by the purpose. Like, let's say we are trying to make a protein, the question we ask why, right? And somebody would say, okay, I'm trying to cure cancer, or somebody would say, oh, okay, I want to make a vaccine, or things like that. But again, things like the thing that I showed, right? I don't even know what kind of function it can do. So
right. Yeah. Good questions. Any other questions? All right. If not, thank you, Dr. Mout. Thank you for your time this morning. Morning for us, I guess, is evening for in India, for this enlightening session. And I really appreciate your time and answering many of my very simple questions, so I can learn a little bit more about what it is. So biology is, or chemistry was the hardest subject for me back in, at the level of higher secondary. I think I barely passed it. So, but I understand the importance of it, how especially as I been thinking about this in the context of computing and how it plays a role to be able to do new things. It just seems so fascinating to me at the intersection of those interdisciplinary ways. So, and thank you again for your time. So,
yeah, thanks. Thank you so much.